Thank you, Electron, and thanks everyone for coming. It's really nice to be able to give a grand rounds. It's almost exactly 2 years after I started in surgical oncology here, and it's funny. I was talking with my father last night, and he said, "This is your first, grand rounds as faculty." I said, "Yeah." He said, "Maybe don't do that thing that you like to do so much." And I said, "What do you mean?" He said, "Infotainment." He said, "Your generation likes that, but not all others do." And so I said, "Well, it's going to be hard to change the way I speak, but at least I can apologize for it up front." So this may be a clash of style. So let's start. Where do we sit with pancreatic cancer? And I think that's what's the crux of this whole talk. And really, it's kind of disappointing because it really hasn't changed much over the past 10 years. But this is a study that was done in 2014 in Cancer Research that is still highly cited because, all right, minor hiccup. Okay, so what's different about pancreatic cancer? And in theory, really, this should be the easiest cancer to target. Very few cancers are so consistent in their molecular driver, and you can see from this graph in terms of different cancers and KRAS mutations, around 90 to 95% of pancreatic cancers are associated with an activating KRAS mutation. And so we have both mouse and human data to show that if we knock out that activated KRAS, most tumors can't continue to survive. But as most of you know, KRAS is often called undruggable. And despite enormous effort in the pharmaceutical industry for decades, we've little to show for it in terms of lives saved. It is worth noting, however, that the FDA has approved a G12C inhibitor in lung cancer, but unfortunately, we can see that in pancreatic cancer, G12C is less than 2%, and we still don't have a G12D, G12V, or G12R inhibitor. There are some in the pipeline, but they're still pretty far away from approval. So what if we take a deeper genomic approach, right? This was the trend, I think, in the 2010s when we sort of learned how to do this whole genome sequencing and got better at it, and were able to put a lot of these data together. And then every couple of years, something would come out in Cell or Nature that says, we took an even deeper sequencing look at pancreatic cancer, and here's what we have to show. Here we can see one of the more highly cited studies. This is the Moffitt phenotypes that's still quoted today of basal versus classical. But really, none of these efforts have resulted in any meaningful treatment changes. So if we put molecular targets on hold, what about early detection? We hear about early detection a lot and how we need it in pancreatic cancer. We need a better way to getting at tumors before they're locally advanced and metastatic. And this is really where probably the closest effort in this is circulating tumor DNA. This is Bert Vogelstein's work. There are a number of companies that have come out like this, CancerSeek, that can look at circulating tumor DNA and get some idea of when there's an early cancer. They're not quite sensitive enough for prime time yet. And mainly, they're not quite specific enough. But while the implementation of a biomarker, just like a better CA19-9 would clearly have clinical use, I think we have to look long-term, right? Even if we are able to have early detection with this cancer, what are we going to do if we can't localize it? And that's a big problem. We can't take out everyone's pancreas because they come in with a high blood test. And so we have to think more about it. And what if we do change people, if we can localize it and we change people from, a metastatic presentation to a early stage presentation, what's the best case scenario? And it's not great. Here, this is a relatively recent study. Now, granted, the outcomes of this study are often cited as not necessarily being representative, but it was a randomized control trial looking at Gemabraxane versus FOLFIRINOX. And this is the best case scenario. These are the resectable patients, right? And even with resection and chemotherapy, and these are the best guns we have for pancreas cancer, it's basically one of two treatments. We see the median survival is less than 2 years. And we see that overall survival in the best case scenario at 5 years is around 20 to 30%, depending on what you look at. So it's sobering, right? Even if we had the best early detection on this stuff, we still can't quite get to a place where it would put it in a cure rate like breast cancer or colon cancer. And so here we are. So when I made this talk, I really, really struggled to get ideas to how to get a room full of surgeons interested in fibroblasts. But really, there's always been excitement in this field, and hopefully you're starting to see some background as to why. We really struggle with molecular targets in pancreatic cancer. It's not that we don't know them, it's just that they're very hard to target. We struggle with chemotherapy and surgery. The chemotherapies we have are very big guns. They're very difficult to give, especially to frail patients. And radiation really doesn't improve survival. That's going to be a controversial statement if you take it outside this room, but there have been a lot of randomized trials on this, and we're really lacking data to show it's efficacious. And so where do we go? What else is unique about pancreatic cancer? And that's where you get to this part, the fibrosis. And this is a typical H&E you see of pancreas cancer. Sorry, trying to get this laser pointer off the screen and it's not wanting to go. There we go. Okay, sorry, this is a typical H&E of pancreatic cancer. And so what do you notice, right? It's a lot of pink, right? There's a whole lot of pink in here. And most of you that remember some histology know that blue is typically a sign of a nucleus. That's a sign of a cell. All this pink is typically fibrosis. And this is pretty unique about pancreatic cancer. And then when you get down and look at what these cells are, you see these red circles up on the right. That, you know, a lot of cells in this microenvironment, in the tumor microenvironment, are actually fibroblasts, up to 80% in some tumors. And so this is what we refer to as the fibrotic stroma, or the mesenchymal component. And we're not the first, certainly, to come up with the idea that this is contributing to the pathogenicity of pancreas cancer. It's been analyzed by most of the large genomic studies. Everyone's known it's a problem. But the question is why? Why is it happening? Why is it specific to pancreas cancer? And why does it seem to contribute to therapeutic resistance? And how do we sort of formulate that into a plan moving forward? And I think I do want to address this, and I promise I'll show data later on to address this, but I think a common clinical misnomer is that this is just pancreatitis, right? There's pancreatitis around a cancer, and this is a very similar process as the benign fibrosis and pancreatitis. And we'll show some data later on that really argues against that. So let's go back to the beginning. And this is going to be some basic science, but I think it's going to be pretty easy to digest. And of course, by the beginning, I mean 2013, when this model really started to solidify about the pancreatic stellate cell. This is a lot of effort from a lot of different labs. I think one of the leaders in the field was Minati Apti, and really developed the stellate cell model. And most people now believe that pancreatic stellate cells are just pancreatic fibroblasts. We don't really see a specific stellate cell phenotype. It was named after that because it looked like a hepatic stellate cell on histology, but it's not really a term that we use much more. We just group it in with pancreatic fibroblasts. We thought at the time that this was just a uniform population, right? That all of these cells in the middle here were just pancreatic fibroblasts, and that when you transition to something like normal pancreas to pancreatitis or pancreatic cancer, it was just this continuous state of activation. You start to lay down matrix, you produce cytokines, you produce growth factors. And so if we think that this is one cell and this is one continuous process, then it really becomes very simple as to what to do about it. And you can either stop the activation or you can kill them, right? And so this is when, in 2014, 2 studies in Cancer Cell came out that really rocked the field, because that was sort of the running thing that we thought was going to happen. We thought that on the top, if we just killed them, that we would improve tumor outcomes. And on the bottom, if we stop the fibrosis, that we would improve tumor outcomes in pancreas cancer. And the reason both of these studies, they literally came out back to back in the same issue of Cancer Cell. The reason most of them are so highly cited is because they agreed with what was going on at the time clinically. Sonic hedgehog inhibitors were a huge promise in pancreas cancer. There were a lot of trials that were devoted to them. Basically, what we found out is if you disable the hedgehog pathway in fibroblasts, you don't get fibrosis. And so that was the promise. And so I just want to take 1 second to look into these 2 models. The first model at the top is mice that spontaneously develop pancreatic cancer. And they're crossed with mice that on a smooth muscle action promoter express thymidine kinase. Why is that important? Because thymidine kinase phosphorylates ganciclovir. And when ganciclovir is phosphorylated intracellularly, it's extremely toxic, right? So it's a way to kill cells that express a certain gene. So this model killed all cells that express smooth muscle actin. Well, that's almost every fibrotic cell in the body, right? It's not ideal. It's not a good model, but it was the best we had at the time. And we thought, even though the body's taking a hit in fibrosis, we're going to hit the tumor, right? And we saw the opposite. Immunosuppression was implicated in this, but really what was happening is I think we were hurting the host more than we were hurting the tumor. But again, this is controversial. And then with the hedgehog inhibitors, the second study at the bottom, so very different model looking at a similar phenomenon. And what they found is that they took this same spontaneous pancreatic cancer model, and this is going to come up a few times, so I just want to take 1 second to explain it. Hopefully, most of you kind of know the Cre-Lox model, but basically, you can produce an enzyme on a specific promoter that then cuts out parts of the genome. And so this is a PDX1 promoter, or any pancreatic progenitor cell. It produces the Cre enzyme, and then it cuts out these spots, the stop codon for KRAS G12D, the stop codon for yellow fluorescent protein. It cuts out sonic hedgehog, so it can't make the ligand, and it cuts out P53. So these are mice that develop pancreatic cancer, and that pancreatic cancer can't express hedgehog. And because of that, you don't get mechanoresponsive activation of fibroblasts in the microenvironment. And what we saw was the opposite of what we expected, right? These mice died very quickly of tumors. These tumors were very poorly differentiated. And so the conclusion of this was really that fibrosis seems to restrain pancreas cancer. And it was the opposite of where the field was going at the time. And I think it surprised a lot of people. And so, what do we do with this information? And this gets tough. So I realize that most of you have no idea what this is from, but you have to be true to yourself when you're making analogies. And so my cousin snuck me into Starship Troopers when I was 12. I'll never forget it. It rocked me. A 12-year-old seeing that movie in the theaters is totally inappropriate. At the time, it was one of the most violent movies ever made. But you never forget it. And I think what I'm trying to get across is it's very easy to go from results like this and just say, we should stop studying fibrosis and pancreas cancer, right? It seems to restrain the tumor. We're doing the wrong thing. We're going to hurt more people if we do this. But really, you have to understand the fibroblasts, right? You have to understand its context. And now we have the tools to do that. We really didn't have the tools when these studies were published. Okay, so what kinds of things do we have to understand? We have to understand how the fibroblast reacts with the microenvironment, right? And it doesn't look like he's here. That's okay. But I think to really understand that and how these studies are designed, we have to understand how immunotherapy works. And for junior residents, this is going to seem super basic. But you have to remember that if anyone's my generation or above, we didn't learn this in med school. This wasn't part of immunology. We had to figure this out on the fly when these therapies started working. And so, I gave grand rounds at my old residency program, and I included this analogy. It was really more as just a dig on Tom Reed. But 2 years later, it's the only thing anyone remembers. So I sort of modified it, and we're going to talk about how checkpoint inhibition works in the context of surgery. As an analogy, it's not going to be perfect, but I think it does help explain the concept. So let's picture everyone's favorite IABAN dress surgeon. Dr. Poltsides is a T cell, okay? Just bear with me for a second. I promise this is going somewhere. So young George Poltsides, it's so hard when I can't see him, but young George Poltsides is coming to Stanford ready to go. He might be a little shy, but he starts to gain experience. And you can picture early T cell activation in this framework. So early activating receptors are upregulated along with inhibitory receptors. And here's where we see this PD-1 receptor, this programmed DEF1. Initially, when a T cell is activated, you get both activation receptors and inhibitory receptors upregulated. Most of us know that. But, despite the inhibitory receptors initially going up, that's okay. He's young, he's ready to go. The activation gives him experience. He's a much more effective surgeon. Now he can do more volume. He's excited, right? So let's say the pancreatic cancer volume doubles, right? It's not a problem. In fact, it's kind of exciting. He's Hopkins trained. He doesn't have little kids yet. It's a dream. He doubles his caseload. He makes a bonus. It's good for everyone. And so let's say the volume continues to double. Well, he still loves to operate, right? But he's getting tired. So he needs to multiply, right? And this is not a good analogy of clonal expansion, but you multiply, right? You get more surgeons, right? Now there's a bunch of us doing the same operation, right? And because there's more volume. And so, we keep going. We're still excited, but we're getting tired. And then the volume doubles again, right? Now people are really getting tired, right? These inhibitory receptors are accumulating. You can't keep going like this. Now we've got families. Now we're married. It's very difficult. And we can't just, it seems like we're not even making a dent in the volume that's coming in. And then it doubles again. And now we're exhausted. We're done. We're not going to do any more pancreas surgery. There's too many cases. And so this is not just a case. The only reason I brought this up is not just to show what exhaustion looks like, right? It's to show the evolutionary reason for these receptors, right? Because let's examine this situation closer. What's the most likely reason for us to have so much more sustained pancreas volume? Well, the most likely reason is because the indication for surgery has become anyone with a pancreas. So now think about T cells, right? They're not made for chronic continued activation. Why? Because what scenario would demand this? If they're attacking something they're not supposed to be attacking, right? So if you have host tissue in your body and T cells decide to keep attacking it, they're going to be activated as long as that tissue is present, right? And that's not good. And so this is the corollary for these inhibitory receptors. This is why we have them. Mice without CTLA4 die of autoimmunity, pretty much autoimmune everything early in life. And so think about this for a second, right? We have T cells, mostly in tumors now, who are just chronically activated. Remember, by the time a tumor gets to be something we can see, it's estimated it's been present for five years, sometimes 10 years, depending on how fast it's growing, right? It's been there a long time. So a T cell reactive to that tumor is almost always exhausted. It's constantly being stimulated. It can't handle the volume that's coming in, and eventually it just gives up. And that's why these immunotherapies were called releasing the brakes on T cells, right? You're suddenly able to take away these inhibitory receptors, and under the right context, you can actually start to eradicate the tumor. And these are the drugs that most of you have heard of. Keytruda and Opdivo are certainly the lion's share of everything. Yervoy was the first one that came out. CTLA4, the work on that was Jim Allison's Nobel Prize. This is a big deal. This really changed the way we saw immunotherapy in cancer. And so to bring it back home, imagine we could now create George clones with no little kids, never get tired, and sort of a religious fervor for taking out the pancreas, right? So let's say that volume's real, right? Let's assume it is cancer and we need to address it, right? Suddenly we have a tool, and that's what a lot of these drugs are doing. They're reinvigorating these T cells to address sort of an evolutionary limitation that was not made for cancer. And that's how checkpoint inhibition works. So where do we go with this? So does checkpoint inhibition work in pancreas cancer? No, not alone. It's been shown time and again. It's not effective. There are a couple reasons proposed for this. We're going to focus on the immune microenvironment within the tumor, right? Because I think something that's cited a lot is there's not enough mutations in pancreas cancer. You're not going to have enough T cells that are specific to it. So now, no matter how much you stimulate those T cells, they're just not there, right? And I think adoptive therapies have sort of shown us that this isn't the case. And these vaccine studies that I'm going to show you in a second really show that that's not the case. I think the T cells, more and more data are coming out that the T cells are there. It's just that the tumor is profoundly suppressive, right? We know that we can give a CAR T cell that can target pancreas cancer pretty well. There are limitations in that, to that concept. But the main problem is we can't get them in the tumor and we can't get them to sustain activation. So even if we know T cells can react to this tumor, as soon as they get into the tumor, they seem to become dysfunctional. And that's something we're trying, that's the whole point of this talk. Why are we looking into this? That's what we're trying to reinvigorate. And so I just want to touch on the vaccines that have come up, because this was a recent New York Times article. I think most people are interested in this topic, and it's not an incredibly complex topic. It's been researched for a long time. But the original pancreatic cancer vaccine was developed by Liz Jaffe in the 90s. This is GVAX. If any of you were involved with the pancreatic cancer vaccine trial, it was probably this one. This came from two patients' cancers in the 90s. They created cell lines. There's a giant GMP bioreactor at Hopkins that propagates these cell lines. And then they get transfected with GM-CSF to attract dendritic cells. Then they get irradiated. Then they get put in the patient, right? And these are just subdermal injections. And they've been really hitting this hard for 30 years. We do find exciting things. It seems to work in mice. It seems to change the immune microenvironment in humans. We do see more tertiary lymphoid structures, more immune cell infiltration after these vaccines, but we haven't really hit that big punch yet. And the most recent study they've done was adding both PD-1, which is an immune checkpoint inhibitor, and urelumab, which is actually a stimulator of an activating receptor on T cells. So sort of a double hit along with the vaccines. And compared to historical controls, on the blue dotted line on the far right there, they were pretty successful, but still not to the point where they're even close to FDA approval. And this vaccine has really been hit on for a long time, and so I think a lot of people thought that it was time to rethink things a little bit, and that's where the New York Times article came out. This is the Memorial study, and we can break this down, very simply, I think. This is very similar to the COVID vaccine, the theory. This is BioNTech's design. It's an RNA vaccine. What are the advantages to an RNA vaccine? It's HLA independent, right? You put the RNA in the cell makes the protein, the cell breaks it down, right? You don't have to worry about making peptides that specifically bind certain HLAs. And so the RNA vaccines can be given to everyone. And what you see is they'll take the patient's tumor out, they'll sequence the tumor. This is a very old strategy. The NIH has been doing it forever. They'll sequence the tumor, they'll sequence the whole blood, right, as a normal correlate. They'll compare the two, and they'll look for differences that might lead to different proteins being expressed, right? And these are your neoantigens, right? A lot of times they're point mutations, right? Just one peptide difference. And you put that onto an RNA vector, and then you stabilize that RNA somehow. This was, really BioNTech's addition to this, because if any of you have gotten the COVID vaccine, if you're someone who reacts to it, it's very different. It's different than any vaccine I've ever gotten. I have to take the next day off. And that's, I usually have a pretty high threshold for doing that, but I'm just useless. And so these vaccines stimulate an enormous innate immune response. And I think that was probably one of the keys to this design. So anyways, they made the vaccine. It was personalized, right? So this is a huge effort. This is a lot to do for each patient, right? Each patient has their own set of mutations with their cancer. The vaccine's personalized. They give it on week 9 after a dose of a PD-L1 blocker. And then they give 8 priming doses. Then the patient gets standard of care adjuvant therapy with FOLFIRINOX. And then they get one more booster after that. And then they were followed. That's the study design, right? So really, it's still the best case scenario. These are the best patients, right? They're resectable patients. They can get 6 months of FOLFIRINOX. So there is a selection bias here compared to what we typically see. But the results are pretty impressive. And if you put them all together, they're not that impressive, right? But they were, the advantage to a neoantigen vaccine is you can actually get some idea of a T cell specific expansion, right? Of an antigen specific T cell expansion, right? Because you know what you're looking for, right? If you have a whole cell line, it's very hard to predict how T cells are going to respond to that. It's very hard to predict the T cells that are going to target that, right? So testing it afterwards is tough, right? Because if you don't see a clinical response, you don't know where to go. But in these, you can test these specific neoantigens ex vivo on patients' T cells. And so what they categorized was non-reactors and reactors, or responders and non-responders. And so it split the group by half, right? And so they put responders in this category where if they re-stimulated T cells with these RNA antigens, then they would respond. And they presumed that there was expansion. And then they would actually do T cell receptor sequencing later on to make sure that these clones that responded before were still present. And that's how they categorize responders. I think the fact that it's split 50-50 is a little suspicious because it's pretty perfect in terms of trying to analyze two groups against each other. But nonetheless, the people they categorized as responders had no recurrences. And this is why it was in the New York Times, right? Because that's still, this is very early follow-up. But for pancreas cancer, that's an achievement. But you can see if you took everyone, it's not great, right? The outcomes are not great, right? The non-responders did not do well at all. And so it does go to patient selection. There's a lot more that needs to be done with this, but I think it was a bright light in a field that has been neglected for a while. Okay, so let's reel it back in, right? I think we need to know about those therapies because we need to know how we're going to look at these cancers in surgical models. And so we come back to cancer-associated fibrosis, and all of this is looking at the interface between fibrosis and immunity. But this is a massive topic, right? This is a whole, micrograph of fibrosis and trying to get at why and what are the consequences. And so when you get something like this, you have to break it down into simpler things, into digestible pieces, right? So I broke it down into 4 different pieces here. This is very subjective, but I do think it captures a lot of the complexity of the topic. So we're going to start with fibroblast heterogeneity. Remember I said before, we all thought this was one continuous process. It was one type of fibroblast responding to basically an anger signal, right? It gets activated, and that activation is both mechanoresponsive, meaning laying down matrix, but also inflammatory. And what we've come to find is that's just not the case, right? There's heterogeneity. Fibroblasts are different phenotypes. They're not all the same. And they differentiate into different cells that respond to different things. And so we have to back up and look at what fibroblasts we're actually targeting. So there's a lot of history here, but I'm going to try and summarize it as quickly as we can. So before the Tubis study in 2017, single-cell RNA sequencing was not really used in this to look at this phenomenon, right? To look at fibroblast heterogeneity. Tubis study, this study from Cold Spring in 2017, was a big deal. It showed that, really we have what they called 3 types of fibroblasts, really 2. The third one's pretty controversial, but the 2 I really want to get across are myofibroblastic, right? Matrix, or inflammatory, cytokines interacting with the immune system, and they're not the same. They're very different. And so this study pointed that out. A whole lot of studies came out afterwards to really get into, okay, well, we're saying there's 2 major categories, but it's got to be more complex now. Now that we can really characterize these things super well, there must be more. And that's where Shannon Turley's atlas came out, and this was only with a few samples on the right side here. But they grouped it into 6 clusters in humans, right? And now you're getting to the subjective aspect of analyzing these data. And the more single-cell RNA sequencing that comes out, the more subjectivity there's going to be in how many definitive clusters or phenotypes there are, right? So eventually, you have to break this down into something digestible from a functional standpoint. And that's what gets pretty hard, right? These 2 studies, it's hard to fit them into the same puzzle. And so, enter Deshka Foster and Michael Janicek. So I am not going to take credit for this study. I was in the right place at the right time to sort of finish the work, but this is Deshka's baby. And so what it did is we used a lot of single-cell technology. So this is RNA sequencing, attack sequencing, which I'll explain a little later. But it really is more of an epigenetic analysis, spatial transcriptomics, codex, a lot of different things to look at heterogeneity in fibroblasts. And I think the result was we came up with a pretty good framework, and it fit with both the Tubis study on the left and the Turley study on the right. And it sort of delineated 3 superclusters, right? This was immunomodulatory, which Deshka gets really upset if we call it inflammatory, mechanoresponsive, but not myofibroblastic, and steady state, which was actually a new cluster, right? You have to incorporate normal tissue, and that's going to come up again and again. There's a steady state-like phenotype that really is present in normal pancreas. And so you can imagine we came up with these 3 clusters. Turley came up with 6 clusters. Tubison came up with 2 clusters, mainly 3 if you consider antigen presentation, but that was a very minor cluster. And so So it prompted this kind of snarky editorial after our manuscript was published in Cancer Cell that said, what's in a name, right? And it gets at how do we make sense of this? You know, what should we do moving forward? How do we categorize these things? I think that our framework is a pretty good compromise between function and specificity. And that's really what you're going for when you're looking at these single cell RNA sequencing studies. So we followed this up with a massive informatic effort. This was led by Michael Janicek, pictured here at the bottom left, with 3 others who lived in his office. You'll recognize one of them. That's Maria, that's John Liu, and that's Serena Jing. They probably lived in Michael's office for, I don't know, 6 months, and looked at every single cell RNA sequencing study in solid tumors. Unbelievable effort. So what this ended up with is over 2,000 samples, over 10 million cells. And to put that in perspective, a typical single cell RNA sequencing experiment is about 10,000 cells. And analyzing that typically takes a pretty nice computer, right? So now you're looking at 10 million cells. And they broke the Stanford supercomputer a couple times, but they were able to get it done. And really what they ended up finding is very similar to what we thought. And this is across over 20 different tumors. This is in mouse and human. This is a huge database of cancer-associated fibroblasts, right? We wanted to be more definitive. What's out there? If we look at different tissue, do the types of fibroblasts change? And we did this with all the stromal cells, not just fibroblasts. But really we get down to, we still can use our global superclusters, right? We still can use this steady state-like phenotype, an inflammatory phenotype, which granted is very diverse, and a mechanoresponsive phenotype. And so it just reinforces that these superclusters are sort of applicable on a global scale. And I really just have to credit the team again. They put a ton of work on this. Maria had never coded before she started this in single cell sequencing. I don't think she had any interest in it. And now she's a pro. And so this was a big deal. This was a big effort for them. They just submitted to Nature. Certainly, I think it's going to get a nice reception. So we talked about this single cell attack sequencing. Why are we doing this, right? Why examine epigenomics at the single cell level in addition to RNA sequencing? And what are you really adding? What are you getting out of it, right? We're at Stanford, so that could be an answer in itself as to why we're doing it, right? The technology was invented here. But it taught me a lot. I had never dealt with it before I came here. I had to learn it on the fly. We do have some experts here, but it's a much more difficult data set to analyze. And how it works is at the very top left here. It's kind of hard to see, and I apologize, but it hits open areas, open regions of DNA, and it cuts up those regions, and then it sequences the areas that are cut up, right? So you end up with peaks. These are very short segments, and they're mapped to the genome. And the idea is, if it's in an open region of DNA, right, it's open for transcription. And so you're getting at how are we regulating gene expression, right? Not just what the gene expression is, but how we're regulating it. And this also gets back to transcription factors. Transcription factors are much more reliable to target than random genes found on single cell gene expression. And pathway analysis is not as nice as you would think. And so I think this does have its place. And I can show you just briefly, this is, again, the people here are amazing. Chunner, I really want to point out, this is Deshka and Dr. Norton sort of started this effort with doing single cell multiome in pancreas cancer. We've done 7 of these now. And this is just a very, very small snippet of that data. But Chunner did her PhD in a similar technology as this. She adapted, looking at attack sequencing incredibly well. I have a lot to say about the residents coming in the lab that are positive, but I truly think Chunner's different. I think, this is, if you listen to her speak about this stuff, I think, she's really someone who is going to make a big impact in this field. And that's when you start realizing that these people are going to rise pretty high above where you are. But it's somewhat flattering, to know that you can mentor them, but it's also certainly humbling. So anyways, when we look at this data, right, we can see the cancer-associated fibroblasts. And, we get some confirmatory markers. There's inflammatory component in red. There's the myofibroblastic component in blue. But really, what's the output of attack sequencing at the single cell level? And it's this thing in the bottom left, right? It's these you get hits, right? These are very small sequences you have to remember that are being targeted. And so really, they just amount to peaks, right? You get peaks along the genome, and peaks correspond to areas that are open. And you can kind of convince yourself that that's what that is when you kind of look at these areas, right? So this is EpCAM. These peaks should be present in single cells that express EpCAM, like epithelial cells, neuroendocrine cells, acinar cells, and it lines up pretty well, right? You see a pretty nice area of gene regulation for EpCAM expressing cells. And so this is nice. The attack data corresponds to the RNA data. We're all happy, but it doesn't tell us much more than that, right? So what do we do with attack data, when it's in a different scenario? And I think this really illustrates how you can use this. And again, this is all Chunner. And I'm sure I'm butchering it in terms of the scientific explanation, but let's take the prototypical fibrosis gene, COL1A1, right? And let's look at it in these different cell types. Because if we would expect to see a pattern like EpCAM, right? You're going to get peaks, right? Where obviously this is an area of regulation here. You're going to get peaks where that cells that express COL1A1, they're going to have. And so what do we see here? We see really only two cells express collagen. We can see myofibroblasts and inflammatory fibroblasts, but we have those big peaks everywhere. So what does this mean, right? How is this gene being regulated if this region is open in every cell type? And the answer is probably, again, this is very preliminary, but you can do this with a couple different genes, and you get the same thing here that we found so far. You zoom out on the genome, right? We often think of promoters and enhancers being very close to genes because that's a very easy, it's a very straightforward framework to think about it. But when we zoom out, right? So again, this is the COL1A1 gene, right? Same thing here, except zoomed way out. What do we see? Well, in cells that express collagen, like myofibroblasts and inflammatory fibroblasts, we see about 50,000 base pairs downstream, specific peaks, right? So what does this mean? This probably means that there's an area of regulation because the linkage is pretty strong between this area of the genome and COL1A1 expression that may be something we don't know about yet, right? And that's important when we're getting to targeting, because oftentimes in these open areas, that's where you get transcription factor binding. And so with attack sequencing, you can do transcription factor footprinting to get an idea of what might bind here. And sure enough, you get some hits, right? And these are new targets, some things that have been associated with fibrosis before, but new areas on the genome. And so this is the kind of thing that single cell attack can give you if you analyze it very carefully. And you would say, well, isn't it much more attractive to do this with something like T cells in cancer? And the answer is yes. And that's why some of these labs at Stanford publish a paper, like a Nature paper every month, looking at T cell exhaustion, because attack is giving us a lot of new insights to that. Okay, fiber analysis. We're going to move quickly now. We've given heterogeneity its due. The fact of the matter is, though, that most of the fibrosis in pancreas cancer is acellular, right? So then we asked, can we learn anything from that matrix itself? And so switching gears to the engineering side, we really asked whether we could develop an algorithm to characterize the type and extent of fibrosis in clinical specimens. The impetus for this came from a general theme in the lab. If you hear Mike talk about it, that if we're going to treat fibrosis, right, we need a better measure of efficacy than just is there fibrosis or not. And so really it took an image processing algorithm to look at a lot of different features of fibers, and you sort of characterize it along this UMAP, and you develop this kind of gestalt that then becomes an official algorithm of how organized are these fibers, right? So the starting place, the trajectory route, is sort of normal tissue. When you have, you don't have a whole lot of fibrosis, the fibers are nice and neat and parallel. And then at the end, you get a much more random assortment of extracellular matrix. And this algorithm was created by Jason and Shamik in the lab, also very talented scientists. They both have just very keen minds for computational stuff. They looked at over 13,000 histological images from 449 patients to come up with an algorithm that consistently approximated matrix architecture, characterized it reasonably well along this spectrum, and then eventually got at, what effects does the architecture itself have on survival? And it was fairly significant. I think some of this goes to tumor grade. We can kind of visualize it that way, but there is more to it. And I'm going to come back to this in a second, but we don't want to look at CAPS in isolation. We can't look at the ECM in isolation if we want to look at immune cell infiltration. And so we really need to be able to look at that in a broader view. And again, this is Jason and Shamik's paper that just came out in Cell Reports Medicine, but they did codex. So think of codex like immunohistochemistry on steroids. You do a 30-plex, image. You can get all these markers in the same image. You can start to characterize not only what cells are infiltrating the tumor, but how close they are to each other, and look at relationships there. I'm going to cut to the end here because we're coming a little short on time, but really the high ECM pseudotime, which means really disorganized extracellular matrix, and the presence of inflammatory fibroblasts were two key components of poor prognosis. There are plenty of others, but obviously that's what we're going to focus on moving forward. And then soluble mediators in the extracellular matrix, right? So we've looked at cellular infiltrate, we've looked at different fibroblast functions, we've looked at the ECM itself. What we have ignored is what are immune cells seeing as they're coming into this matrix, right? As they're migrating in. And that's a really hard concept to get at because immunohistochemistry doesn't typically work for secreted soluble mediators. They're oftentimes too small. And you really can't see it with gene expression, right? Because if it's secreted into the matrix, it's not necessarily being expressed anymore. And so our approach was really to look at what's a condition with a similar matrix, right? And that's where chronic pancreatitis comes up. And so is there really a difference between the fibrosis in chronic pancreatitis and the fibrosis in pancreas cancer? They look very similar, right? It's a whole lot of pink. It's a whole lot of extracellular matrix fibers. And the way we did this is very simple. We went back to the basics, right? We took the specimen, we ground it up, and we looked at cytokine secretion with a bead assay. And the Luminex has been around forever. It's a very easy test to run. It's very cheap now. But when you grind up these samples, we did 159 pancreatic surgical specimens. The orange is chronic pancreatitis. The yellow is pancreatic cancer. The blue is benign pancreas that was resected for some other reason. Obviously, it's never completely normal pancreas. But you can see there's a whole lot more red at the top of the screen in pancreas cancer than in pancreatitis and benign pancreas. And actually, pancreatitis approximates pretty closely to benign pancreas in terms of the soluble mediator milieu or the secreted factors within the matrix. So there's a different type of inflammation going on in pancreatic cancer, so much so that you can take most of these cytokines and they become very close to being diagnostic. Now, obviously, that's not clinically useful because we have pretty good ways of biopsying it now. At the time, it was exciting because EUS was just much less reliable in a lot of centers. But certainly, it's been sidelined since. This was I was really encouraged to do this by Steve Hughes. I should certainly credit him. He was very interested in chronic pancreatitis. He still is. I think I still carry some of that with me. And I think it's just really interesting to look at differences between pancreatitis and pancreatic cancer, because comparing pancreatic cancer to normal pancreas is just not helpphel. It's so different. It's just so different. The histology is night and day. And so if you're looking for targets, this used to be the old method of doing things like phage display and things like that. You would grind up normal pancreas, you would grind up pancreatic cancer, and you'd look for differences, and you get extracellular matrix. And so I think if you're gonna compare it to a benign condition, it has to be pancreatitis. Okay, so summary thus far. So 3 dominant CAP phenotypes, significant diversity within each one, but we can make these global clusters. The extracellular matrix can be prognostic, the cellular infiltration can be prognostic, but you have to be able to consistently measure it. And the inflammatory pattern is really a departure from benign fibrosis in pancreatic cancer. All right, so where do we go? And this is a ton of information. I didn't want to present my dissertation because it's a long time ago, but this is still the line of work we're doing. And more or less what we showed is that when we convert a mechanoresponsive fibroblast to an inflammatory fibroblast in culture. That conversion seems to be dependent on the MyD88 pathway. So MyD88 is downstream of toll-like receptors. It's downstream of IL-1. It's a very commonly used innate immune pathway. We can convert these fibroblasts in vitro, and these are human fibroblasts from pancreas cancer specimens. We can convert them in vitro to this inflammatory phenotype. The inflammatory phenotype secretes a lot of cytokines, namely IL-1 initiated things like IL-6 and IL-8. Fibroblasts don't get a lot of credit for secreting cytokines, but they actually can secrete a lot of chemokines that attract neutrophils, on par with myeloid cells. And when we do that, when we convert them to these inflammatory fibroblasts, remember they tend to cluster around immune cells in the tumor, we actually find that they're profoundly suppressive of T cell functions. Once they're converted, T cells cannot function in their presence. They can't proliferate on bead stimulation assays, they can't skew to Th1, they can't respond to interferon gamma, and they can't kill tumor. And we showed all that. I encourage anyone who wants to read these papers. It's a win-win for me, but it's a lot to read. And so you're gonna have to trust me for now. And we also showed that these fibroblasts, the cytokines they secrete, particularly IL-8, led to profound cancer-associated cachexia. And this was a big project when I was in graduate school with one of our collaborators, now a close friend, Andy Judge, who's a muscle biologist. And we did a lot of different rectus muscle specimens from patients undergoing surgery for pancreas cancer, and developed a couple great mouse models to look at this. He's still funded to do this, to look at what fibroblasts produce in the tumor that actually leads to cachexia. And so this is what I was awarded the Damon Runyon for. This line of work, we can briefly summarize sort of where it's come. You know, the main obstacle I hit in year one here is how do you demonstrate this how do you examine this in vivo, right? How do we look at fibroblast MyD88 signaling in vivo? Because MyD88 signaling is it's probably, you know, more commonly known in myeloid cells, right? And so how do you separate the two? And so you need a fibroblast specific genetic model, and you can't do that with the transgenics, right? Because anytime you manipulate and by the transgenics, I mean the mice that spontaneously form pancreas cancer, anytime, any cell-specific genetic manipulation in those mice has to be on the pancreatic cancer cells. And so you can't manipulate fibroblasts at the same time. So we can do the old style where we just injected tumor cells into the pancreas, but the problem with that is they pretty much form sarcomas. You know, they're like 80% tumor cells. There's very little cellular infiltration. There's very little cellular diversity. And so even though we can manipulate fibroblasts, if there's no fibroblasts in the tumor, then it's pretty hard to study. And if there's no T cells in the tumor, then it's hard to combine it with immune checkpoint blockade and take anything away from it. So in comes the new model that we've been spending a lot of time working on. The idea here is we embed the cancer cells in a collagen gel. We give them some matrix. We let them come to equilibrium in culture. We let them get back to exponential growth phase. We let the things they secrete, the chemokines, the growth factors, embed itself into the matrix, and then we implant it. So rather than dissociating the cells and just injecting them in the tumor where most of them are going to die and some of them are going to take up residence, but then it takes them a while to sort of reestablish their microenvironment, we sort of we implant them with their microenvironment. And this is the procedure. It's very easy to almost minimally invasive implant something in a mouse pancreas, just a very small left subcostal incision, and the pancreas falls right out. And so we see they develop nice pancreas tumors. We see some fibrosis. We see nice neovascularization. We see tertiary lymphoid structures, but don't take my word for it. Look at the single cell data. So this is single cell data at the top left from the collagen gel implant model. This is 7 different tumors combined into one. The diversity was pretty consistent versus the cell line injection model, right? And what do you see? You see mostly cancer cells in red in the cell line injection model. You see a ton of different cells in the collagen gel model. We were really thrilled with this result. And when you look at things that might have to do with immune checkpoint blockade, you see that T cells are actually a prominent cell type in the collagen gel model. You see things like CTLA4, like PD1, PDL1, and different myeloid cell subtypes. You don't see any of this in the cell line injection. And so it's really hard to examine these things in the cell line injection. So what happens when we actually look at it? So when we knock out MyD88 in a fibroblast-specific manner, this is a COL1A1 CRE, and then we do the collagen gel implants, we can see that the tumors grow slower, and we can also see that T cells are more activated in the tumor microenvironment. also more exhausted. And so, and this is, this is 4 experimental mice. This is 3 controls that we did single cell RNA sequencing on. And we can see across the board more exhaustion markers in T cells in the experimental group and smaller tumors. And so it suggests maybe that this is a good strategy to do with immune checkpoint blockade. And we do see stromal cell changes. We see actually more fibroblast infiltration in the Col1 MyD88 flocks. The fibroblasts are not inflammatory. They're mostly steady state and mechanoresponsive, which is sort of what we expected. We didn't necessarily expect to see more. I'm not going to go through this slide in detail, but certainly happy to answer questions. So conclusions: fibroblast innate immune signaling pathways contribute to suppression of T cell function in pancreas cancer, and combinatorial therapies to address chronic inflammation from persistent MyD88 pathway stimulation may hold promise in immune checkpoint blockade. There are excellent ongoing projects. I don't have time really to highlight them, but I do want to talk about just one real quick, the PancVax gel that we're starting up here. This came from an idea that what if we can't resect the entire tumor? Is it time for sort of a regional immunotherapy? Liz had this, already established vaccine for the pancreas tumors in mice. There was a STING agonist, Adavax, and peptides. We took it into an incomplete resection model. So you lift off most of the tumor, you leave immunized systemically. So despite the operative trauma, we actually see immunization after surgery when the implantable vaccine is delivered. And so we are hoping to get this American Cancer Society grant the second time around to continue this work with Obi Chowdhury here to develop the right hydrogel. I don't really have time to go over the others. Deshka is going to make $1 million. And we are doing some great stuff with uterine leiomyosarcoma. This is with Bern and Beatrice, Maria, Sarah, where we're actually looking at these cells in a much deeper genomic level than has been previously done. If you've seen one of these women, it's a horrible story. They get peritoneal recurrence, and they're often young women, and they just undergo 5 or 6 laparotomies until you just can't do anymore. Bern treats these patients a lot here. I really didn't have experience with them before I came here, because I think Gynonc, really just gives them palliative care. And so this is really a tumor that needs targets. There's not even transcriptomic data publicly available, because that's how far behind we are with it, because it's rare. Memorial has a genomic database, so we know mutations, but we don't really know how these cells behave and what makes them different. We found 3 major phenotypes. We put them into a drug discovery pharmacogenomic algorithm. It's the first one of its kind that actually looks at single cells and the transcriptome of each cell and how likely it is to respond to drugs based on known pharmacogenomic databases, which are actually pretty large now. We saw that, and you can see here that they respond. A lot of our quality control checks were satisfied, right? The agents we currently use on them, gemcitabine, 5-FU-docetaxel, cisplatin, etoposide, crystine, all of those are predicted to work on the cancer as a whole. Steroids are predicted to work on myeloid cells. ALK inhibitors are predicted to work on lymphocytes. VEGF inhibitors are predicted to work on endothelial cells. It checks out. I think it's a really powerful algorithm. And then, I think the take home is not a single drug works on all 3 cancer cell phenotypes. So combinatorial therapies are going to be really important. And I think this is why we never see even close to a complete response with chemo in these patients. And so that's kind of the take home there. Again, there's so much I can say. It takes a village to make one of these jobs work. And there's just been an unbelievable amount of support here. I met my wife in the lab. We got married. You can see that's our son and dog when they were the same age, much younger. Now they're older here. The newest member of our family is going to be a sequencer. Thank you to everyone who's contributed to that. And then everyone in Mike's lab, all the residents coming in. Deshka is an unbelievable human being. Both her and Chunner, they're going to rise way higher than I am. And it's just such a pleasure to work with everyone here. People are excited. My partners are incredible. you cannot run a lab if you don't have supportive senior partners, and everyone's just been unbelievably good about helping me when I needed clinically and mentoring me. And so I couldn't be in a better spot. I couldn't be happier. Thank you.